So you got 20,000 genes in a cell. All of your cells have that 20,000 genes, but most of them are going to be turned off.
A cell doesn't need them, like a liver cell only needs what a liver cell needs.
Yeah, that's right. So you want your liver cell to keep all those other genes turned off. And that's what makes it a liver cell, because those genes are turned off, right? Only liver cell genes are turned on. And so even though every cell in your body shares the same genome, different cell types have different epigenomes. And what is the job of DNA? It is really just to code for proteins. So if you keep the DNA tightly coiled and not expose the gene for transcription, then that gene won't be expressed. The protein won't be made.
So you're just not letting that strand be read.
Exactly. So how does it do that? There's proteins called histones, which the DNA wraps all around, really coils around it. And then there's also methyl groups. They attach to your genome and they kind of accumulate over time. So your chromosomes get more methylated throughout your life.
And that's attached to the histones or to the DNA itself?
So it's attached to the histones, which help it to coil the DNA. And similarly, there's another molecule called acetyl, which also attaches to the histones, and that actually helps to unwind it, to expose the genes, right? So it's this kind of balancing act between the methyls and the acetyls. The metaphor for the methyls, which I heard is a scientist called David Sinclair out of Harvard, and he wrote a book on this called Lifespan. And he talked about the methyls as being like plaque on teeth. So it just accumulates over time. And now I don't know how they do this, but they can actually look to see how methylated your chromosomes are, and they can estimate your age. And they also know sort of what it would look like at end of life, and they can sort of anticipate when you might die.
By the rate of accumulation of methyl groups, they can predict when you're going to die from age.
It's like a clock. Right? Obviously not if you're going to get hit by a bus or something like that, but so
that's scary.
It's pretty amazing. So what I've been learning about lately on this topic is that there's a group of proteins that help to keep your epigenome intact.
So just those genes that are being expressed.
That's right. Keep the ones closed up that are supposed to be. Right? Their job is to maintain the epigenome. They code for an enzyme that removes acetyl groups.
I see. So in the moving of acetyl groups, it'll keep that histone closed up.
Exactly. That's their main job. But if there's damage to the chromosomes, they are also involved in repair. And if they have to go repair, they literally leave their post managing the epigenome. They go to repair the broken chromosomes, and then they go back. But when they go back, they don't always go back in exactly the same place they were. So every time they come back, they're like just a little bit off of where they were originally, which can just cause some slight changes in your epigenome. So what this latest research seems to be on this is that the cell has this way of maintaining the epigenome, but when they go to fix the chromosomes and they go back, there's that slight change, and it's causing changes in the epigenome, which eventually can lead to genes being expressed that shouldn't be expressed. And when that happens, the cell loses its identity, right? Like that liver cell we mentioned. Now it's maybe no longer really a liver cell.
Yeah, it's functioning haywire.
Right. So those proteins that preserve that epigenome, they stay there and do their job when the body is under a little bit of stress. And apparently it's a bit of a survival mechanism. So when times are tough, the cells evolve to sort of hunker down and just maintain that epigenome.
So that it doesn't start to express things it shouldn't.
Yeah. So it's sort of like maybe getting a little bit ahead of ourselves here, but thinking about what that means.